Bioconversion Group, which is doing a lot of process optimization for, in this particular case, mainly organic acids, which could be, and you might know that, further polymerized into then logically biopolymers. And this is one of the main interests of our group. So just to give you a brief overview of that, what we are doing in our institute. The long history, also the institute, but after the reunification in 92, reestablished, and with a lot of new topics besides the classical agricultural engineering, which was the history of the institute. Nowadays, you find the word bioeconomy in the title of our institute. It means we are covering more or less the entire range of food and non-food applications for that, what we expect for the next years, decades. I will come back to that during my presentation. So the so-called research program areas in our institute are illustrated here. And of course, I'm not talking about animals or about the crop production itself. I'm also not talking about food, which is a important element of the bioeconomy for sure. But if we come to that part, the biomaterials and the so-called residue management, this is more or less the topic which is mainly covered by my former working group, Bioconversion, as we are trying to use different residues, biogenic materials, to convert them into, not in the most cases, not directly into the biomaterials, but first into the chemicals like the organic acids, which then can be subsequently processed into materials. So this is more or less what we are doing. And a bit more in detail, one example for using different types of waste, what you can see here on top, bio waste, what you can have in your brown bin in front of your house, or other bio waste or paper waste, cardboard, whatever we could have. What Thomas already mentioned, which is in so many cases not already used for other purpose. And therefore, we are looking for have those materials in our processes to make that what is illustrated on the right-hand side in the top of the circle. So that means we are doing the hydrolysis of those materials, we are running the fermentation, and we do the downstream processing to get the pure chemicals in the end of the process. And two of our main products are lactic acid and succinic acid. So as you might know, lactic acid could be processed into PLA, polylactic acid, and succinic acid in PBS, polybutylene succinate. These are the both polymers we are looking for. And just to give you a brief overview, you know that all from the metabolic activities of our microbes, what we can get from, but I marked here again the two products what we are working on, the lactate and the succinate. And of course, you can go to all those forecasts and outlooks for the market. What we see for the last years, it's the sector is growing, but as you might know also, especially here in Germany or also in Europe, we are a bit lacking on the implementation of those technologies. So we are very good in R&D, I guess, but it's not that easy to bring that in the scale what is necessary to make the bioeconomy as a real contribution to the economy. So this is what we have to achieve. But I think we all are working on this, and we have all the same the same interest. So another example for those different waste what we were using from the left, so from the kitchen, from the food industry, from the organic fraction of municipal solid waste. So not the brown bin, this is the very dirty black bin, what we are collecting, all the waste or garden waste. And our idea is to avoid the what is still in place, the burning, or also to make just compost out of it, which is um, of course necessary to have compost for the gardening and things like this. But we have so many different materials what we could burn or give to the composting process that we think that we should go directly with part of those raw materials into our fermentation systems and come up with a product in the end. The second of our products is here an example where we were using sugar beet pulp, which is a residue from the sugar mill, as you can imagine. And we are trying to exchange the former gray, dirty process on the right-hand side here, the petrochemical route, by a green route with fermentation and using also the byproduct in this process. I just give you an idea about some main parameters. I'm not going to go into the details. The only one what I would like to mention here with those graphs, that of course, in view of the process analytical techniques, we are interested all the time in having quick information about our main parameters, which are on the one hand, of course, the substrates, the sugars on the one hand, the nutrients, and for sure the final product. In some cases, we have by releasing some inhibitory compounds out of the waste material, also phenolic compounds or acetic acid, things like that, which is disturbing our microbes. And this would be also in our interest to get quick information about such substances. You all know the four main steps of those bioprocesses, and here again a nice picture of our pilot plant, which we put into operation in 2006. It means next year we are celebrating 20 years, sorry, anniversary. And this was really very important in that what I was mentioning already, the scaling up of processes. And you see on the right-hand side of that picture, our biggest fermenter, 450 liter, connected with all the pumps and equipment like the hollow fiber membranes to run it also in a continuous mode. So I just highlighted still the two, in my opinion, main challenges we still have: the pre-treatment of those dirty materials, complex and dirty, and in the end, separation and purification of our products, since this is, of course, influenced by the raw materials itself, what we are starting with. And I think we have to do a lot of work in order to make solutions for the industry. So here I just like to summarize that what bioengineers do all the day in the lab or in other facilities. So as you can see from the title of that slide, that the most important thing is that we are quicker and cheaper, to say it in simple words. So I was also already talking about the pre-treatment, where we want to have as much as we can sugars, but as less as we can the inhibitory compounds. We could apply mixed cultures or multi-substrate processes to make it more flexible. Of course, the space-time yield is one of the main performance parameters of our process, and we have to develop advanced strategies like SSF or continuous mode cultivation. And I was also talking about the downstream processing for the recovery of our product. So in the end, all of that, what we are doing with that measures before is to establish optimal process conditions for our cells, for our microbes, for our enzymes, whatever. And for that, we can develop application-specific hardware systems, the bioreactor itself, and of course, very important here, the process analytical techniques, which could support and should support us in getting the right information, preferably online, which is not the case in so many applications. So I was already talking about the scaling up, which is, in my opinion, one of the most challenging task. And as you might know, of course, it's not that easy, like in that funny picture here, that we just needed to make the glasses a bit bigger. In reality, we have to work really hard on bringing things not only in a scale like we do in our pilot plant with 100 or 500 liters. We have to achieve 100 cubic meter, 200 cubic meter, whatever. So in this is not that easy. And we don't have, unfortunately, I have to say, not so many examples already in place where it is realized for those, especially waste using processes in order to make products out of it. So why it is also that is well known, but it's not easy to overcome, since the bigger the liquid volume we are operating, the bigger is yeah the tendency that we have inhomogeneities inside of our reactor. And of course, all the different process conditions are not the same for our microbes, and we have to do that it is close to the ideal conditions for the microbes. So just to come to the end, this is what we are doing all the time in our facilities, starting with the strain, with optimization in the lab scale, going the scale up until the pilot scale. What we are not doing, I have to point out, we are not modifying the strains. We are just doing the process optimization, not the strains itself. And with a brief summary of having that, what I call the triangle, the feedstocks, the processing, and the products, with the different things I was already touching during my talk, the advanced strategies, for instance, where we look for partners to provide us with feedstocks, as Thomas mentioned before, and to have, in the best case, continuous mode processes in order to increase the overall process performance. And the increase of TRL is very important, as you all might know from the different fundings, especially the European funding, which is a very important topic for your projects, to get higher TRLs and not like 10 or 20 years before making just the R&D in the lab scale. And I was also talking about the downstream processing, which is an important part of the entire process. And with that, I thank my, I have to say, former team, which is, of course, still working on the same topics and all the funding what we got. And in case you are interested in having any collaboration with us, so I would be happy to do it by myself or to bring you in contact with my team. So thank you very much for your attention.
Thank you very much, Joachim, for your great talk and keeping the time. Is there any question? Holger, please.
Yeah, thanks for the interesting talk. I heard something like you build the own bioreactors. Is that for small scale or is it for big scale?
No, we're not building the bioreactors by ourselves. We
You
are
are
doing the
designing.
process optimization.
Ah, okay, good. Thanks. Sorry.
Are there further questions? Yes, please, Mr. Pandey.
Oh, thanks for the talk. Just wanted to ask, what does the LCA for such studies look like? I mean, what are they really feasible, or yeah, that's a simple question.
Sorry.
Just wanted to know what does the life cycle assessment look for such processes, right? So ultimately, whether this actually makes sense to do or not, because ultimately, you can only commercialize it if it actually makes sense, right?
Of course. I mean, I was talking about the hurdles in order to implement those technology in the industry, and maybe 20 years ago, it was mainly due to the cost and the economy, which was not feasible. But as you all know, for the last years, it was more and more important to also demonstrate the sustainability of those processes, the CO2 footprint. And of course, you are right. With those relatively young technology, it's not easy just to snip with your fingers and bring that process in reality in a few years. So we are really working for more than 20 years, or I myself. And to be honest, I was expecting this time, 2003, when I was starting this work, that I would see better results much sooner than I have could see right now. Of course, we are in contact with industry, with companies directly, contract research, and they have interest in those processes. But in the end, I mean, if we don't have the product in the market and we don't see the industry doing that, it's clear there are some still lacks, not only the economy, but also the sustainability. And this is what we have to achieve, I guess.
Thank you very much. I'll probably take up some more questions later with you. Thank you.
Please feel free.